Good morning, all. We'll give people a couple minutes to sign in. I'll try to keep this fairly short and sweet, even though we're recording it, just so it's not like if people can't make it because they're out, driving around, dealing with the conditions, and can't view it until later. It'll be something that we can pick up again on Wednesday's class and, hopefully not skip a beat. So we'll give people until maybe 10:27, maybe 10:28 to log in, and then we'll go ahead and get started. All right, so let's go ahead and get started. Before we do, does anybody have any questions about the course so far? We've only had one lecture and then we jumped right into a workshop, which was probably pretty good that I changed the schedule to put a microscope workshop, first week, because now we're going to lose some of that microscope time since we won't have lab today. So are there any questions about anything? Okay, cool. We'll see if I can still remember how to switch my screen over. So give me a second and we'll go ahead and get started. If you have a question, feel free to go ahead and interject just because I'm not sure. I'm not used to looking for like the raised hand anymore. So maybe raise your hand first. And if I don't answer within like a few seconds, go ahead and feel free to cut in. We should have plenty of time to answer questions during the lecture. So that's no issue. Or put it in the chat and I'll try to check that by the end of lecture and be able to answer any question there if you're feeling a little shy and don't want to interject directly, although you're welcome to. So let me see here. Can everybody see that? Give me some thumbs up or something in the chat.
Yeah.
All right, cool. Thank you. All right, I'm going to see if I can minimize my part of this other screen here so I can see all the slides. We'll go ahead and get started. So on Wednesday last week, we started to go into some of the basic characteristics of life. And life is really one of those questions. If I ask you to define, like, what is life? It's kind of like tricky, hard to define. And there's actually quite a few characteristics that living things have to have in order to be alive. So we're going to look into some of the more specific aspects of those characteristics that we went over in class on Wednesday today. And a big part of that has to do with cells. And that's why we started out with the workshop on Friday, starting to look at the differences between prokaryotes and eukaryotes. And those different types of cells are going to go about doing things in slightly different ways, but they're also going to have a lot of similarities and a lot of things in common in order to go about their metabolism, in order to be able to live and reproduce and fulfill all of the roles that they need to do in biology. And a big part of that is how they manage to manipulate their internal environment and keep it separate from the external environment, because obviously what happens in your cells requires a lot of chemistry and a lot of really specific physical conditions. Like there has to be a certain amount of water in your cytoplasm. That's why hydration is important, right? It has to be a certain pH for a lot of reactions to occur. And also it has to be within a certain temperature range. So there's all these specific things that need to be, happening just the right way in order for cells to do their thing. And a big part of that is going to be a big part of that big part of manipulating that internal environment is going to have to do with the outer membrane of the cells, the cell membranes, and how cells can actually bring water in or, kick waste products out. So that's what we're going to be talking about today in lecture. And that's what our workshop on Friday is going to be based on. On Wednesday in lecture, we can spend a little bit of time kind of brushing up on this if necessary, and because it's a little bit of a different format than what we're used to. After about a year and a half of being mostly on Zoom, now we're mostly back in person. So we can allot some time Wednesday to going into this a bit more. And our main topic Wednesday will be starting cell division. So that'll be our week in a nutshell. So let's get started on these, like, why are cell membranes so important? Well, obviously maintaining that outer environment and keeping those physical characteristics different from the external environment is what allows cells to really be able to function. So the cell membranes themselves are composed of these really cool molecules called phospholipids. And these phospholipids are really unique in a lot of ways. They have one side, if you look at this bigger diagram here on the left, that they basically have two main parts that have really different properties. So they have a hydrophilic phosphate head. In other words, hydrophilic, it's really attracted to water. It's capable of being soluble in water. And then we have this tail of hydrophobic fatty acids. So we will be able to basically repel water. Like that side of the molecule is going to be totally different. So you've got this one molecule with two sides that have totally different properties. And what's really cool is when you put this in water, what's going to happen? Is it going to dissolve or is it going to just hang out? Well, it's not going to really do either of those things. The side that's going to be attracted to the water molecules, they're going to tend to point one way. And the hydrophobic acid tails, if you have a group of these molecules all together, they can self-organize to point the other way. And this can lead to some really cool structures that look almost like, they've been built or designed, but really they only set up this way just because of those electrostatic interactions on the hydrophilic phosphate head and the hydrophobic fatty acid tail. So the simplest type of structure that you can have is this micelle, which is on the left in this bigger diagram here. And that's basically just around, you can think of it almost as like a clump of these phospholipid molecules. But because of that repulsion of the hydrophobic fatty acid tails, if you have a group of these in water, this clump will self-arrange itself so that all of the fatty acid tails point inward. So you see how all those tails are pointed towards the middle of that micelle? That basically creates this really symmetrical spherical object, which looks almost like, it would have had to be built, right, in order to be so symmetrical and so well organized. But really, all it is the electrostatic interactions of those phospholipid molecules with the water around it, which is pretty cool because symmetry just arises from, the natural properties of these molecules. But the micelle is the absolute simplest type of structure that you can have from these interactions. So liposomes are a little bit more complicated. And now if you look at the liposomes, now we have something that looks a little bit more like a cell, right? Because in the micelle, there's no open cavity. There's no interior space. It's just all the fatty acid tails kind of pointing at each other. In the liposome, you have this alternating pattern where on the outside of the liposome, you have all these phospholipid molecules with the tails pointed inward away from the water, which makes sense, right? Because just like with the micelle, they're repelled by the water. So the tails that are repelled face away from them, they face inward. But in the liposome, we actually have a situation where when that layer was forming, somehow some water got in the middle. So we're going to have some of those phospholipid molecules that are actually going to face away from that interior water, away from that inner cavity. So now we have basically this hollow spherical structure. And once we have that, we have something that looks a lot like a cell, right? And if we look at this diagram on the right, if we make it a little bit bigger, you can see that cavity on the inside. The more phospholipid molecules you add, the bigger it's going to get and the bigger the interior cavity is going to get. And when you have water on both the outside and the inside, those phospholipid molecules are going to self-organize so that they're staggered, where every other one is going to be pointed inside at that inner cavity or outside away from that inner cavity because there's water on both sides. So different molecules are going to be repelled so that they face in opposite directions. And they will actually self-organize so that they're staggered, so that every other one faces the opposite direction. And this is where you get that bilayer sheet that lower figure on the left-hand side kind of shows you in a little bit more detail, where on each side you have a layer of phospholipid molecules that are oriented in opposite directions. So basically these structures, these are held together not by chemical bonds, but by electrostatic interactions. So the phospholipid molecule is held together with chemical bonds, right? Because it's a molecule. But these phospholipid layers that form a micelle or a liposome, they aren't rigid. They're not really, really hard. So these molecules can kind of slide around. They're flexible, almost like a soap bubble, which actually is not chemically that different from these micelles and liposomes. So liposomes actually have cell-like behavior. They're these self-organizing complexes that have some lifelike qualities, right? It's almost like if you go out into the environment, we can actually find these types of molecules that form naturally, no biology necessary. They just self-organize in environments that have water and basically the components of that phospholipid molecule. So phosphorus, carbon, and hydrogen, basically. And those are all elements that are really, really common in the Earth's environment. So it shouldn't really be that surprising that we see these types of things in nature quite a bit. But going from, if we go back one slide, from something that just looks like little bubbles of oil or lipids, floating on the surface of water, to something that's like a liposome or even like a larger, more cell-like structure, that'll take some special conditions, right? But you can kind of see how the natural environment around us actually creates cell-like structures, even when there's no biology present. And some of these lifelike qualities that they have are that they can provide a locally segregated environment. So that inner cavity of the liposome, there's an opportunity for the chemistry that occurs there to be different and sheltered from the outside environment. So these boundaries can also allow the selective adsorption of simple organic molecules from the surrounding medium. In other words, it can attract them and bring them into this area and make that inner cavity, make the chemistry that's going on there even more unique. Also, they can grow and divide. And if you've ever like just basically done the dishes and you had made something that's a little bit oily, oil and water, like how do they mix? They look like this because the oil molecules they're like the fatty acid tail here, the hydrophobic fatty acid tail. They're going to be repelled by the water and they're going to form, these globs. Probably all of you here in the lecture, have seen this kind of thing before. And that's basically how liposomes form. So we have a few origins. We have a few hypotheses on the origin of life, because now we've seen that, from the abiotic, in other words, the non-living, non-biological components that are present everywhere in the environment, we actually can get some structures that actually resemble primitive cells in some ways. So this idea, one of the hypotheses for the origin of life is that of chemical evolution. And in the first step of chemical evolution, we see small organic molecules such as amino acids and nucleotides being formed in the environment from simpler elements and compounds. And these are the same types of simple organic molecules that we saw on Wednesday could actually be created in the lab, like in the Miller-Urey experiment. We actually saw that a huge assortment of amino acids were produced, just from the abiotic components of what the scientists at that time had thought was the atmosphere of the early Earth. The joining of these small molecules into larger molecules or macromolecules can result in proteins and nucleic acids, because amino acids, when they start to chemically bond to one another, they can form these larger proteins, right? Proteins are basically polymers consisting of amino acids. And nucleotides do the same thing. They can link together to form nucleic acids and eventually very large polymers, very large macromolecules such as DNA and RNA. The packaging of these molecules into these structures that we can term as protobionts, which means something like, it means technically like early biont, like something that resembles a cell but doesn't quite have all those characteristics of life just yet. So protobionts were probably, complicated liposomes, basically droplets of oil almost with membranes that were kind of shaped like bubbles that were able to maintain an internal environment that was different from their surroundings. And they allowed chemical reactions between molecules to be carried on inside the membrane of those protobionts in an environment that was a little bit different from the outside, and maybe over time, quite a bit different from what was going on in the outer environment. And this is the precursor. This was the beginning of what we would later see resemble something more similar to the biochemistry that happens in cells that we see around us now. And then finally, the development of protobionts that can actually reproduce molecules like DNA and RNA that are then able to replicate themselves and make inheritance possible. There's also another piece of this. When we start talking about biology, once we have a living cell, now we're talking about biological evolution, right? Before we get up to an object that has all the characteristics of life, we're talking about chemical evolution. But once we have an object that has all those qualities, in other words, a cell, now we're under the domain of biological evolution. And this brings us to the heterotroph hypothesis. And in a nutshell, part of our story here that we'll talk about in class is how some of these cells, when we take a survey of different cells across all of the domains of life, we're going to see that there are big differences in structure and function, and we could see that just in the workshop on Friday. There were these early cells that were anaerobic heterotrophs. In other words, anaerobic, they did not need oxygen to survive. They were heterotrophs, which means that they have to get their energy from somewhere else. They have to get it from another living cell. They can't get it from the environment. Plants that photosynthesize are called autotrophs because they can take up energy from the environment and they can fix it into biological molecules that they can use as an energy source. and that heterotrophs can use as an energy source. So when you think heterotrophs, think of that Greek root hetero, which means different. Just remember that if you're a heterotroph, you have to get your energy from a different source. You can't just pull it from the environment. If you're an autotroph, think auto, like automatic. You can just hang out and basically absorb energy and use that to fix it into molecules like glucose that you can use as an energy source later. And that's what plants do when they photosynthesize. So some of these early anaerobic heterotrophs started to evolve, and they began to release carbon dioxide into the environment. Well, photosynthetic anaerobic autotrophs, such as cyanobacteria, that carbon dioxide actually can be used to make the glucose that is the energy storage molecule that photosynthesis yields. So for photosynthesis, plants or cyanobacteria or other photosynthetic organisms, they take water and carbon dioxide and they use sunlight as the energy source, and they combine those two molecules into glucose. In other words, basically a food source that you can use respiration that utilizes oxygen to break down. But the byproduct of that reaction for a plant is oxygen itself. And this is one of the reasons why, even like, kindergarten, pre-K students, at least it was kind of drilled into me that, animals, when we exhale, we breathe out carbon dioxide, but plants breathe in carbon dioxide. If you want to kind of anthropomorphize plants a little bit and think of them as something that breathes, really, they don't breathe in the same way we do. They have little tiny pores in the leaves called stomata that allow gases to enter the leaf. And they will bring in carbon dioxide that way, combine it with water during photosynthesis to create that glucose. The waste product of that reaction is going to be oxygen. So you can kind of think of plants as breathing in carbon dioxide and breathing out oxygen, even though obviously they don't have lungs, so they're not actually breathing. They're just letting these gases diffuse in and out of their leaf surfaces. So kind of building that story a little bit from these first few cells, aerobic autotrophs can use oxygen, and it turns out that gives them more energy for more growth than some of the other cells that are present, in this early Earth environment. And this is going to be a competitive advantage, because if you have more energy, then you can do more things. And if you can do more things, you're going to be a stronger competitor. So eventually, as time goes on, some of these cells start to become more and more complicated and more specialized at doing certain things. And eukaryotes end up evolving from prokaryotes. And when we say eukaryotes, if you remember from our workshop on Friday, eukaryotes are cells that have a nucleus. And a nucleus is basically the command center of the cell. So the nucleus is regulating a lot of the activity that occurs in the cell. It's sending signals to the organelles. It's receiving signals from beyond the cell membrane, which we'll talk about a little bit later in this lecture. It's basically running the show. Prokaryotes don't have a nucleus. So their command and control structure, as humans, we kind of have this view that if it's a simpler system, it's more primitive. Well, that's not necessarily the case, but prokaryotes generally have a more simple setup to their cells, and they don't have basically like a command and control center like the nucleus. They can accomplish the same task, but the process is basically, it's more structured into individual parts within the cell itself. There's no centralized command and control center with the nucleus, which, in some situations might be advantageous in its own right. Obviously, it works pretty well, and they're still competitive with eukaryotes because they still exist in nature, right? If it wasn't efficient and the eukaryotes were, so much more superior as competitors, the prokaryotes would have died out a long time ago. But clearly that's not the case, because there's still plenty of them around. Eventually, as some of these cells became more and more complicated, they started to congregate in colonies, and they would, over time, these colonies became one organism that consisted of multiple cells. And from here, we start to get into some of the familiar organisms that we see around us, like, for example, ourselves, right? We don't just consist of one type of cell. We are an organism with trillions of cells and dozens, if not hundreds or thousands of different individual cell types that we can classify with much different functions. Some of the big obvious groups being, blood cells, like cells that have to do with your immune system, bone cells, brain cells, nerve cells, cardiac cells, which is, basically a specialized type of muscle cell that's in your heart. We can go on and on. So to step back and just look a little bit more at these phospholipid molecules, because this is going to be really important for telling our story about what happens with these cells. Just a little bit more detail here. This is just a simple version of a phospholipid molecule. Don't worry, you don't need to in this class, you won't have to reproduce a sketch of like a phospholipid molecule with every little atom that's present, but you should be able to identify the different parts of this molecule. For example, if I gave you this diagram on an exam or on a quiz, and I said, label the polar head, the glycerol backbone, and the fatty acid chain, I might copy and paste this diagram and say A, B, and C, and fill in the term for A, B, and C. You should be able to identify the polar head, the glycerol backbone that connects the polar head, with the fatty acid tail or fatty acid chain that comprises the tail. And then just a different sort of structure here for our more visual learners, just to show it to you from a few different angles. This is more what it looks like in real life right here. So you've got your polar group, your polar head, your phosphate and glycerol parts that basically cement together the polar head and the fatty acid tail. And if you can identify those things, then you are going to be well prepared for any assessment that you see. But you're also going to be able, if you can identify those things, it's not just rote memorization, right? Because if you can memorize those things, then you can understand why the phospholipid molecules arrange themselves the way they do in these cell membranes. Because if we have, with this lower picture right here on the right, if we have an aqueous, in other words, water-rich solution on the exterior and an aqueous solution on the interior, then that, we've got this molecule here. One side is polar and attracted to water, and the other side is non-polar and it's actually repelled by water. So you're going to have molecules, you're going to have phospholipid molecules that are going to face away from each direction. In other words, you're going to have a bilayer, because on the interior, you're going to have all of those polar heads facing the inside and all the tails facing away. And on the exterior, it's going to be the opposite, right? The tails are going to be facing the interior, to get away from the exterior water, and the heads are going to be facing the water that they're attracted to. So if you can just memorize that structure and which side is polar and attracted to water and which side is non-polar and repelled by the water, you can basically know how to build a phospholipid membrane from scratch, and you'll know why the membrane self-organizes the way that it does. And this brings us right into the fluid mosaic model of the cell membrane. So with this, we'll look at a little bit more detail on the membrane itself, because it's not just as simple as phospholipid molecules. We actually have proteins that can be going, they can actually stretch from the outside to the inside. And we actually call these transmembrane proteins because they cross the entire width of that membrane through the bilayer. On top of these proteins, almost like antennas, we have these carbohydrate chains, and calling them antennas, they kind of look like antennas, like kind of waving around there, waiting for a signal. That's actually a really good metaphor, because that's exactly what they do. They can be used to sense the environment around the cell, and the cell can use that to change what it's doing on the interior to better match up with and respond to what's going on outside of the cell itself. These carbohydrate chains can also be used to communicate from cell to cell. Cells can release molecules that will chemically react with these carbohydrate chains, change them somehow, and then send a signal, from the outside of the cell membrane to the inside without having to send a molecule directly through the bilayer. So the fluid mosaic model is basically going to be the topic of our workshop on Friday. You will be kind of sending signals from one side of the cell, from the exterior to the interior and back. But instead of using something that's shaped exactly like these proteins, these carbohydrate chains, we're going to be using candy. So in some cases, you will be able to eat your workshop. Although I will tell you that we will be using nuts for some of these molecules, and I would not eat any of the nuts. Like, we'll be using pecans for at least one of these molecules that we're simulating, and the pecans have been in the lab for a while, so I would hold off on eating those. But something that you're opening up, like a box of like those little hearts, that's been sealed and just bought last Thursday at the store, probably okay. But it's always good to make sure with your peer leader that what you're eating is safe to eat. So the three main components of cell membranes are going to be three of those big, actually two of those big categories of macromolecules we mentioned last Wednesday, and phospholipids, so which actually fall into that third category of lipids. So phospholipids, which actually compose the majority of that structure, carbohydrates, which can be on the surface of the cell membrane, and proteins, which can actually be within that membrane. And the proteins can actually act as gateways or doorways. And I will leave this link here. I won't go into it right now, but it's basically a motion picture of molecules going in and out. And we can actually watch that in class on Wednesday if you'd like, but I'm not sure if I'd be able to get it to work right here on the video. So I'm going to just move on. We actually have a couple videos that I was going to play in lecture. One of them, I think actually one or two of them I embedded directly into the slideshow. So I might see if I can get them to work, but if they don't work, quick and easy, I'll just post the direct links on Canvas and you can look at them after lecture. So just to show you the scale. So some of the diagrams we've been looking at before, and if we go back, let's look at the liposomes here. So we see that, in this simple stylized drawing, the phospholipid bilayer is pretty wide, in terms of like the scale of the rest of the object, right? Well, for an actual cell, it's actually rather thin and tiny. It's like paper thin, right? So this is a cell. This is a scanning electron micrograph of a cell. So it's a real life photo of a cell membrane of a cell. And you can see the cell membrane is actually very, very thin and tiny compared to the cell. This cell is actually roughly round. And you can see we're zoomed in so far that you can't even see the curvature of the cell. It basically looks like a straight diagonal line. So just to show you how thin the cell membrane truly is relative to the rest of the cell. So what affects how many substances or which substances can move across the plasma membrane? Well, there's going to be three main qualities of any molecule that will allow it to enter. The first one is size. And this one's pretty obvious, right? Because if we look back at this structure here, we can see that even though this is a stylized picture of a phospholipid bilayer, you can kind of imagine that these molecules, they aren't a perfect fit together, right? If you have a really small molecule, like let's say hydrogen, just a proton, the smallest atom there is, right? Or maybe oxygen, you can probably sneak through this bilayer. So if you are a small enough atom or molecule, you're going to be able to diffuse through that membrane, depending on a few other qualities such as the polarity, like what is your charge, because we're going to have literally some of these proteins that might act like a gatekeeper, because these proteins can act like literal gates, and they might open or close or allow you to pass through them if you're too big just to pass through the phospholipid bilayer based on your charge. So this brings us to two different types of movement across the cell membrane. And we're going to call this movement transport. So passive transport is when we move from high concentrations to low concentrations. And this incorporates just simple diffusion. In other words, small molecules that I mentioned earlier that don't have a really big charge. If it's a molecule and it's not really polar, for example, water is H2O. It's a pretty small molecule, but it has a really strong polarity to it. The oxygen tends to be, really, really negative, and the hydrogens tend to be positive that are on each side of the molecule. So you have one side of the molecule that's really negative, the other side that's positive. And that disparity in charge might block it from just going through the membrane unless, it's helped through in some way. So oxygen and carbon dioxide, they're small molecules that don't have that problem. In most cases, they can readily diffuse through the cell membrane. Another thing that we can add, and basically this is just simple diffusion. Also, movement of substances from a region of high to low concentration. And this incorporates osmosis. And osmosis is the specific case of moving water. We can actually move water across a membrane as long as it's moving along that concentration gradient from high to low. Facilitated diffusion occurs when we have movement by those channel proteins. So those proteins that we saw earlier that span that phospholipid bilayer, they can actually assist as well. So these carrier proteins, these channel proteins can actually open. And in some cases, we have carrier proteins, which can basically leave the cell and go out beyond the cell and grab onto a certain ion or a certain molecule and grab it and bring it into the cell. And those are called carrier proteins. And these can assist also movement from high to low concentration. And this is why it's called passive, because movement from high to low concentration is just a basic law of the universe, right? Energy does the same thing. Matter and energy, which really are, two faces of the same coin, which maybe you talked about a little bit in Psi 100. Both matter and energy move from regions of high concentration to low concentration, always, unless, they're interfered with in some way. And before we get into some of that interference, just to kind of show you how some of these channel proteins can assist with the movement of water, they can kind of like flex or move to the side or open or close or change their shape a little bit based on the presence of different charges. Or one thing that can influence the presence of different charges is the pH. So if we have a lot of H+ ions, a lot of protons, then we're going to have a lower pH, and that can actually push and pull on some of the structure, like the little tiny like bumps and dimples on the proteins, because in real life, proteins might look like they're kind of these curved structures like this. But if we zoom in on them, they're going to look almost like they're bumpy, because there's going to be bumpy or almost like they're clumpy, and they're going to have this really complex three-dimensional shape. And ions that have charges on them are going to interact with the molecules that form those bumps or clumps and kind of change their conformation a little bit and possibly open or close this protein, this channel, basically. But now if we're looking at a situation where we can sort of mess with that simple diffusion, that situation where we just have, which is basically the default in nature, where we move from high concentrations to low concentrations. Well, if we input energy, we can actually do the opposite. We can kind of go against that flow. We can kind of go upstream against that normal current of simple diffusion. And this is called active transport. And this is when we move from low to high concentration, which is, in other words, is against the gradient, against the flow. And we can only do this by inputting more energy. So if you think about swimming in like a river or a stream, if you just sit there and do nothing, if you're passive, you're going to go with the current, right? But if you're going to swim against the current, you have to input a lot of energy, right? And that's pretty much the same thing that's going on with these ions. If you are a cell and for some chemical process, you have to move ions, against the flow, from high concentration, or sorry, from low concentration to high concentration, then you're going to have to input energy to do that, almost like you're pushing them against the current, right? So there's a few different ways to do this. Carrier proteins can move molecules against a concentration gradient. Proton pumps can actually move protons like into a volume so that they become really, really concentrated, and this will basically change the pH for a cell. And you can also use sodium and potassium ions because they are really important for signaling and for a lot of other cellular processes. And here's another YouTube video, which, give me a second. Nope, this isn't one of the ones I directly embedded. Let's be a little bit adventurous and see if I can get this to work. And hopefully my screen will stay viewable to you. Chime in and let me know if it doesn't. Can everybody still see it?
Special Business, the no-nonsense way for your business to stay connected. Get the fastest internet speeds in the nation.
Recall that there are two types of cellular transport, passive and active. Passive requires no added energy, but active does require added energy.
Sorry, did somebody just say something?
Yeah, we can't see the video.
Oh, no, I'm sorry. Well, let me see if I can change how I'm sharing the screen. Oops, that's the chat. Okay, now I think you'll be able to see it. Everybody good?
Yeah, it's good.
Okay, one thing that happens sometimes though, and this might only happen on the recording for Zoom if people watch it later, sometimes the volume doesn't come through. So let me know if the volume doesn't work for you guys.
Recall that there are two types of cellular transport, passive and active. Passive requires no added energy, but active does require added energy. Active transport also moves from an area of low concentration to an area of high concentration or up the concentration gradient. This is the opposite of passive transport, and because it's more difficult, it needs the energy molecule called ATP, which stands for adenosine triphosphate, because there's an adenosine molecule and 3 phosphate molecules. There are 3 types of active transport: membrane pumps, endocytosis, and exocytosis. Let's look at membrane pumps first. Membrane pumps are just carrier proteins that move substances from areas of low concentration to areas of high concentration. Let's look at the sodium-potassium pump to see how it works. Outside the cell, there's a high concentration of sodium ion, or Na+. The brackets represent the word concentration. Inside the cell, there's a high concentration of potassium ion, or K+. The cell has ion channels that will allow these ions to diffuse and reach equilibrium, but the cell doesn't actually want that. It wants to keep a difference in concentration. So the sodium-potassium pump will force more sodium out and more potassium in all the time. Let's see how this works. First, 3 sodium ions from the cytosol bind to the protein, and a phosphate group from ATP breaks off in the carrier protein. This provides energy for the shape of the protein to change, and the sodium ions can be released into the outside of the cell. Now, 2 potassium will enter the pump, which causes the phosphate to break off and return the pump to its original shape and release the potassium into the cytosol. Again, sodium will diffuse into the cell on its own, and potassium will diffuse out. The pump forces the sodium to the outside the cell and the potassium to be inside the cell. Another way that cells do active transport is through vesicle movements, either endocytosis or exocytosis. Endocytosis brings items into the cell, and exocytosis pushes them out. Endocytosis process by which cells ingest external fluid, macromolecules, or other large particles, even other cells. We can be more specific about what materials undergo endocytosis. Phagocytosis is where large particles or whole cells are ingested, and penocytosis is when solutes or fluids are ingested. You can think of phagocytosis as cell eating and penocytosis as cell drinking. Here you can see the way it works. First, the cell membrane begins to pinch around the substances, and eventually it forms a vesicle, which can carry the substance to any part of the cell. Exocytosis is basically the opposite of endocytosis. It's a process by which the substance is released from a cell through a vesicle that transports it to the cell surface and fuses with the cell membrane. A vesicle can merge with the cell membrane because they're both made of phospholipids. The layers press into each other, and the phospholipids rearrange a little so that they can open up their contents to the outside of the cell. Again, this requires energy, so it's classified as one of the types of active transport. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on Twitter at SciencePet.
Okay, let's get back into this now. Okay, so sorry about that. That video is quite helpful. There are some really good resources online. When I learned this material, it was as a visual learner myself, it was really difficult and took a lot of effort. I think if I had some of these videos, I would have been able to develop an intuition for it a lot more rapidly and learn it and remember it a lot better from the start. So we have these resources now. Why not use them, right? Because if we're talking about a really dynamic process with a lot of literally moving pieces, then, you know, videos are the way to go. So just to kind of review a few things, you know, active transport versus passive transport. Passive transport, you know, is basically just diffusion from high to low concentrations. It doesn't require energy to occur. And facilitated diffusion, even if it gets help from certain components of the cell membrane, if no energy is expended that is needed for that to happen, then it's still passive transport, right? So this is something that a lot of students get wrong, is that they think facilitated diffusion. Well, if it's facilitated, you know, the cell's helping it along somehow, so it has to be active transport. But that's not the case, because if it's some component of the cell membrane, like a channel protein that doesn't need to open and close, it can just let specific things through, then it doesn't require energy. So it's still passive transport. But if it's something like we just saw on the video, where you need ATP to bind to that protein and then the right molecule to bind to it for it to open and close, that's a different story, right? It's going to require active transport because the ATP is going to be playing a role and inputting some energy into that protein so that it can change its shape to open and close. So just kind of a quick review here. We don't need to go over this in depth. These are just a little bit more detailed examples of what happens with sodium and potassium pumps. You don't need to remember all these details. It's just to kind of help you develop an intuition on what this process actually looks like in real life to make these pumps open and close. And basically, we have ATP that are going to bind to the protein, and then we're going to have one of the molecules, one of the ions of interest, in this case, sodium or potassium, bind to different sites on that protein to open it, to open that protein, or close it to let it out, or to let potassium in. So basically, this protein here in this multi-step process to let either sodium out or potassium in, we call that a pump, because that's basically what it's doing. It's pumping sodium out and it's pumping potassium in. So we call that a sodium potassium pump. And that's one of the simpler proteins that are used in active transport that help different ions get through the cell membrane, because ions like potassium and sodium, they're not going to be able to just kind of sneak through the bilayer. They have to have specific points of entry if they're going to get in. So these carrier proteins are capable not only of letting them in, but also moving them in against the concentration gradient. So just to kind of look at something that the video also spoiled a little bit, endocytosis and exocytosis. In endocytosis, the cell basically forms like a little dimple, and eventually it's going to wall this off, and this little like cavity can actually move through the phospholipid bilayer. And once it reaches the interior cell membrane of that bilayer, it can release its contents into the cell. So exocytosis does the exact opposite. It's basically cells, it takes a lot of times waste products, and it can release those waste products. It can basically secrete them outside of the cell. And for both of these processes, the way that this happens is that the phospholipid bilayers can actually kind of like change their shape. Like when you got this dimple forming, and eventually it pinches itself off to form this hollow cavity. And then the membrane actually reforms on the surface here, which is pretty cool. So you can basically move something through the membrane without actually, piercing the membrane or breaking the membrane. The surface kind of reforms behind it. So here's a video I tried to directly embed. Let's see if we can get it to work here. This might also have an issue with the sound. So just chime in if that's the case. And if necessary, I'll click the link and switch over to YouTube.
Cells also use energy to transfer materials in bulk, but this time by forming membranous sacs that hold their contents under wraps. In a process called endocytosis, part of the surface membrane encloses the material, forming a sac which brings the contents into the cell's interior. In the opposite process, called exocytosis, the sac moves through the cytoplasm to the membrane, fuses with it, then releases the contents.
I like that video because it looks like a video and sounds like a video that we'd watch on one of those big boxy TVs that when I was a kid on movie day, they'd like wheel in on a giant cart like in middle school. The sound effects are also kind of similar. I don't know how they come up with the soundtrack for some of those old science videos, but they're kind of nostalgic. So let's watch this video as well, and I think this will do it for the videos.
What is receptor-mediated endocytosis? The following animation will describe the process of receptor-mediated endocytosis. To show what happens during receptor-mediated endocytosis, first, let's draw our cell membrane. Note that the cytoplasm is on the bottom and the extracellular fluid is on the top. This means that the inside of the cell is on the bottom and everything outside of the cell is on the top of the page. In the extracellular fluid, there are solutes and particles which are floating around the fluid. These are called ligands. The idea behind receptor-mediated phagocytosis is that there are receptors on the surface of the cell which are specific for certain types of particles. This means that they will only attach to certain particles and leave the other particles alone. When a particle is detected in the extracellular fluid, the ligand attaches to the receptor. The receptor then migrates to an area which is coated in a bristle-like substance called clarithin. Clarithin helps the cell's own membrane form a budding pouch, also known as a vesicle. The vesicle containing the particles are transported to the inside of the cell for processing, while the clarithin and receptors are recycled and take their place back on the membrane of the cell, ready for the next round of receptor-mediated endocytosis. Thanks for watching. We hope you found this video helpful. Be sure to like and subscribe for new videos.
All right, bear with me while I quickly go back to the slide we were on. Okay, so a real life photo of this happening using an SEM, scanning electron micrograph, is basically here's what it looks like. So you can actually see the phospholipid bilayer actually kind of forming that dimple, right, and expanding the boundary around something that the cell is trying to take in and starting to reform over it. And you can actually see the bilayer starting to reform on this example right here. And in this cell, you can actually see some areas where it has already happened. So this is a cell that they caught moving quite a bit of material in, or, since this isn't in motion, some of these, I mean, all of them, for all we know, could be actually moving out rather than in, right? Without motion and without seeing what direction it's going, we can't tell for sure if it's endocytosis or exocytosis. So it's pretty cool that the cell membrane can actually, it's got this trick up its sleeve where it can bring material in without action and then reform itself behind that little bubble without actually breaking itself open. So that video kind of led into, we called it receptor mediated endocytosis because it leads into one of our next points that we want to talk about, which is how cells communicate, right? I mentioned earlier when we saw that slide with the surface of the plasma membrane, we had the phospholipid bilayer and we had the channel protein, but we also had some carbohydrates, if you remember, some carbohydrates chains that were kind of hanging out, that were kind of waving around, that were almost like antennas, right? Well, that's exactly the sort of receptor that can bind some of these free floating molecules or ions that are called the ligands that were mentioned in the video to actually send a signal based on what's happening outside the cell, inside the cell. So we start with these ligands, and the ligands are basically just a molecule that's going to be bound to something, in this case the receptor. So we've got that happening right here. In real life, no, it doesn't look like a monkey wrench grappling a hexagon. The shape is actually way more complex. It's actually like something that's real. It looks almost like a blob. And the receptor, the ligand might look like a blob in some cases if it's a complex biological molecule, and the receptor will have the perfect shape to actually match up with the blob. And so it'll be like pretty much a mirror image so that they fit together like a lock and a key. And that way the receptor can only respond, to a certain blob or a certain molecule, right? And this way it doesn't get its signals crossed. So once that happens, once you've got that receptor ligand binding, it actually sends a signal. It creates a chemical reaction that releases molecules from the inside of the receptor, the part of the receptor rather that's inside the cell membrane, to other molecules that get released into the cytoplasm. And these secondary messengers can then go to different places in the cell, whether it's the nucleus or other organelles or just the cytoplasm itself, and basically send that signal where it needs to go. In the case of the nucleus, if it's something that's going to impact how the cell operates, we're going to actually see changes in gene expression as that nucleus actually cranks out orders for what to do in response to the signal that comes through. And then just another picture. I like to include a lot of pictures because, like I said, I'm a visual learner, and that really helps me out. So hopefully it helps a lot of you as well. We've got that message molecule basically that binds to the receptor. And again, the receptors often are shaped in a really particular way. So it's like a lock in a key. And basically the receptors are really, really specific for receiving, certain messages. And once that happens, it creates a series of chemical reactions. And we've got these secondary messengers that are released into the cytoplasm. And that's what sends the signals forth. And that signal could be, just about anything. It could be something that says, okay, make the inside of the cell more acidic so we can create a certain compound, to release into the environment. Or it could actually be a signal that tells the cell to start growing and dividing. And in a nutshell, this is how cells communicate. When they're going from cell to cell, if it has to be a signal that needs to be really, really rapid, like let's say a wasp, is flying close to your face and you want to swipe away at it, to keep yourself safe from being stung, well, you're going to have to be sending signals to your muscles, right, to operate in a certain way. And in that case, we have nerve cells that have these gap junctions that can really rapidly send an electrical current through these cells that are all linked. So basically, you've got this interconnection via the gap junctions where there is basically an ionic fluid, usually rich in those potassium and sodium ions that you saw earlier. And once, as you know from Sci 100, or any chemistry class you might have had in high school or here at St. Rose, if you have a solution that's loaded with ions, it can conduct a current, right? So you can actually use these gap junctions to send an electrical current to your muscles to tell them how to move and to swat that wasp away. In a plant, obviously plants don't have those types of cells, right? So they have a different sort of mechanism. They have these structures called plasmodesmata, where the cell walls line up. They're these little tiny passageways that match up on cell walls that face each other. And you can almost think of them as like little highways because they're open all the time. And the cell membranes that are inside these cell walls can actually, kind of swell or shrink to open or close them to a larger extent. But these plasmodesmata basically allow ions and signaling compounds, especially, to move around the body of the plant. So even though animal cells and plant cells, and both animal tissue and plant tissue, which contains lots of different types of cells, even though they're a lot different, it's kind of cool how, even though they're so much different, they have such similar mechanisms. And this is an actual SEM photo of plasmodesmata linking different cells inside of the plants. And that's all I have for today. So before I wrap up, I will end the screen share and kind of go back to all of you. It was kind of meant to be, I actually wanted it to only go until probably like 11:15 today, but when I have the screen going, when I've got my screen shared, I can't keep track of time, so I'm 12 minutes over. But I wanted it to be a little bit shorter so it wasn't as imposing today as, because people are going to have to be, I think we have 11 people here, or we've had 11 people for most of the lecture. So a lot of people are going to be viewing this as a recording, and I want to keep everybody up to speed. So is there anything I can go into more depth on before we meet again on Wednesday? Okay, well, one note I will leave you with is don't be intimidated by this. I know it looks really complicated. Every year I have students say that, they didn't really get a good handle on the material in this lecture until they did the workshop, because in the workshop, you're actually going to be manipulating this system, obviously, in the form of candy. But you'll be mimicking active and passive transport and channel proteins and signal transduction and all that cool stuff. And once you do that, I think with your peer leaders, it'll make a lot more sense. I did see we have one question in the chat. Does a cell wall always have a cell membrane? So let me change that question around a little bit. So all cells have cell membranes, but not all cells have cell walls. So cell walls actually are what plants have, right? And that's something that you probably remember from, from K through 12. I think I learned that really, really low, like maybe even in like 3rd or 4th grade. But some bacteria can also have cell walls. I did hear somebody say, I don't know if it was a peer leader or a student, but I heard somebody say in workshop last Friday that only plants have cell walls. That's not true. Some bacteria, some microbes have cell walls. Plants have cell walls, and that's the one we think of the most because that's the organism that we see the most that have cell walls. But fungi also can have cell walls, and they're not plants. So some microbes, some bacteria, plants and close relatives of plants like certain cyanobacteria, And algae, they can have cell walls, fungi can have cell walls. So I think that's a better way to put it. So I'm still learning everybody's names and faces. So BA, I'm not sure who BA is, but I hope that answers your question. Any others? Cool. So it takes a little while for the video to be converted into a form I can upload. So probably in like an hour or so, I will post the recording on Canvas. So it'll be a little while before people can view it. I'll probably get a few questions, wondering where it is. It will take a little while to get it up there, but it should be up very soon if you'd like to review anything, or if a classmate that couldn't be with us live reaches out to ask, it'll be up as soon as it finishes converting and I can upload it. All right. So with that, if there's no more questions, I will see everybody on Wednesday morning. Be safe out there.
Thank you. Have a good rest of your day, everyone.
Thank you.
You're welcome. Bye.